c'est changement de salle. Je peux pas projeter parce que j'ai pas internet, ça se connecte pas. J'ai pas d'internet. It doesn't connect to internet. I don't have réseau.
You're projecting through this computer, no?
Yeah, which is everything is local except
Everything
is local except for the program that it's online. It's online. Online it should work. It's just I cannot connect with my phone or with my computer to the online website where the correct program is.
So let's announce it first now, but then also after the
Absolutely.
roundtable as well.
Yeah.
A reminder.
Okay. Welcome back, everybody. So before we announce the next speaker, I have an important announcement for the workshops in the afternoon. So most of you registered to the workshops. We don't have a list, we're not going to check names, just go to the workshop that you are registered to. If you haven't registered to a workshop, there's still place so for the first session, workshop 1 or 2, there's still space in workshop 2. For the second session, there is still space in workshop 3. And for the third session, there is still space in workshop 6. So another important information is regarding the rooms. So we adjusted there was a modification in the rooms. So it's not anymore what is written in your program. So please bear with me. The online version that you should find is correct. So if you are going to workshop 1 about intellectual property, you should stay here in this room. If you go in the workshop 2 about patent license is on salle 2, so the room number 2, which is upstairs, close to where we had lunch. For the second round of workshop, if you go through workshop 3, the jargon of innovation, you go to room 2 on the first floor. And if you go for the workshop 4, integrated overview of innovation ecosystem in France, it's here in this room. Third batch, if you go for the workshop 5, market overview for researchers, is in this room here, while startup creation workshop 6 is in room 2. I think the online program is correct. And then I will let Ariel introduce the next speaker.
Welcome back, everyone. The future is here. I think that's sort of the message for the next speaker. The future in which researchers can actually think and design and not spend 2 years building one vector, but actually thinking about much more complex systems and have biofoundries do the work for them is here. And nothing signifies this more probably than Ginkgo Bionetworks, a young startup that started with iGEMers, so there are iGEMers here, they know this, and is now the most successful synthetic biology company in the world. And I'm very happy that Jake, who spent many years here in Paris and helped actually mentor, teach, evangelize synthetic biology among students here in Paris, and now back in Boston, give his talk, Synthetic Biology in the Foundry Era. Thanks, Jake.
Hi, everybody. Hi, everybody. Great to be here. Great to be here in this the coveted post-lunch spot. I think we've all got the sleepies. I'm very excited to be here, addressing what I'm pretty sure is the galactic parliament from Star Wars. In this building, we have a lot of talent assembled here today. I think with your help, the Republic will defeat the evil empire, certainly. My name is Jake. I come from Ginkgo Bioworks. I'm going to vamp for a minute here while the slides get assembled. We heard earlier this morning about this vision for building a biotech ecosystem in Paris, rivaling the one in Boston. And I love this vision. I absolutely love it. I think it's a beautiful vision. I'd love to make it happen. I will talk a little bit about myself, a little bit about my biography. So I come from Boston. I've seen it. don't tell them I said this. Maybe that's the evil empire. Don't tell them I said that. I'm American by birth. I'm also French. So I lived in Paris for 11 years. I've seen the Paris biotech ecosystem. I've seen how powerful it is, what it can do, how much talent there is here, if we can just unlock it. 2 years ago, I moved back to Boston, back to the United States, to work for Ginkgo Bioworks, which is a synthetic biology company. So it's a synthetic biology company, it's a platform company. My motivation in working at Ginkgo is the same as the motivation that I had as an academic for many years, working in citizen science and in more accessible biotechnologies. My motivation is that the world needs more biology. So we're at this moment where climate, human health, food insecurity, a lot of the biggest problems that we're facing in the world have biological solutions. All right. I'm trying to think of a joke that I can make about how technology doesn't work, but I can't think of one. And the people in this room, they see that. They see that a lot of the biggest problems in the world have biotechnology-driven solutions. Ginkgo is a horizontal platform for cell programming and for synthetic biology. And I think that we are in a position to make the tools for engineering biology more accessible to more people, including hopefully a lot of people in this room and a lot of people in the Paris ecosystem. My job here at Ginkgo is inspired. Oh, I have to point at this. Oh, that's me. There we go. Okay, so I said that one. We're moving on. World needs more biology. Okay, we covered that. Okay, here we go. Okay, good. I'm glad this one is oh, good. We're going to leave this one on the screen for a while. Okay, thank you. My role at Ginkgo is called Developer Evangelist. And so it is inspired by this beautiful man. This is Steve Ballmer. Steve Ballmer, for the younger people in the room. He used to be the CEO of Microsoft, famous for getting really, really excited about developers. So if you're a platform company like Microsoft, you live or die based on the success of the people who build on top of your platform. And so Steve Ballmer knew that. So he went out all the time and was just yelling, developers, 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 developers. You've got to get excited. You have to love the developers. You have to put them in a position to win and succeed. You have to give them the resources that they need to succeed. So at Ginkgo, the vision is similar. We're a biotechnology platform company. People in this room are the developers, and so we have to love you. We have to give you what you need to succeed, and that's what I'm here to try to do today. We're here to talk about the bioconvergence. And in particular, the way that the bioconvergence changes everything. And so here I'm gonna play the role of the super enthusiastic American, telling you about how, innovation is gonna change everything. We're gonna disrupt everything, right? Nothing's ever gonna be the same again, right? There I go, okay. I'll try to be as concrete as possible about the ways in which the bioconvergence changes everything in this talk. It changes how Ginkgo Bioworks works. It is the reason why Ginkgo Bioworks exists. It changes what we can build with biology. It changes how we design R&D projects, particularly the role of AI lately is really changing the flow, the structure of R&D projects. It's changing who can build with biology, what it takes to be a synthetic biology founder, and it's changing where innovation happens. And in particular, I think it's opening up a lot of possibilities, particularly in an ecosystem like Paris, because the bioconvergence plays to a lot of Paris's strengths as a biotechnology research cluster. So the bioconvergence changes how Ginkgo Bioworks works. What's Ginkgo Bioworks? We are a horizontal platform company for cell programming and biotech R&D. So we do synthetic biology as a service. People come to us because they want a cell line engineered, for example, some sort of synthetic biology R&D project. We create a microbe for them or some other DNA design. We license that microbe to them, and then they use it to create their product. We started in 2008. We went public in 2021 after raising a bunch of money. Now we've got, the last time I checked, there's about 1,200 bioworkers, including hundreds of synthetic biologists, process engineers, data scientists. So we're the biggest synthetic biology company in the platform in the world. We have partnered across the entire bioeconomy. So every industry that biology touches, you can find companies that have selected Ginkgo to power their R&D platforms. And so that includes a lot of companies in the therapeutic space that you could see on the top. It also includes a lot of companies outside of therapeutics, so food and chemicals. We work in industrial bioprocesses. We work in materials, as well as sort of living therapeutics and the entire bio-based and biological therapeutics. So we've partners with companies across all of those industries. And you might ask, well, why is that possible? How is that possible, right? To be in a horizontal platform in an industry that is traditionally much more vertically integrated. And the answer is on that computer right now. But, oh, there it is. The reason is that because biology is fundamentally programmable. So the thesis behind a horizontal platform company, a foundry, like Ginkgo's, is that DNA is fundamentally one technology. It's possible to get really good at DNA programming, and that knowledge, that know-how, transfers to all of the many, many other applications that are enabled by DNA programming. This is the thesis behind the company, right? So DNA is essentially one thing. You can have a company like Ginkgo that handles the DNA stuff for many different partners who handle the product side of building with biology. So, what makes it work? Okay, so I think the first, the most obvious answer is the physical infrastructure in the foundry, which is represented on this slide by some blurry lines. But let me tell you, in the real world, the foundry looks awesome. Like there's like robots everywhere in this picture. If you saw it, it's like amazing. We've invested about $500 million worth of in infrastructure, in robotics infrastructure, and so that's only the most obvious part. There's also a lot of software infrastructure that goes into making all those robots work together, but it enables the possibility to build with biology at scale. The second component is the code base. So the code base is something that should be very dear to the heart of the synthetic biologists in the room. The code base is reusable biological parts. It's organisms, it's pieces of DNA, like promoters, that are useful across many different projects. It's data that we accumulate over the course of doing many different projects. So this is our code base. This is what enables us to get better at programming cells every time we do it. The foundry and the code base together make a flywheel. So we have economics of scale in the foundry. Big infrastructure reduces the cost of unit operations. That lets us work on many different projects for many different partners, build up our code base, and then reinvest in that capacity to create this flywheel where every time we work with a new partner, we get better, and the costs per unit operation can go down. The only way that this works is because we can partner with developers, with people doing other R&D projects. If we were only making our own projects in-house, if we were only doing our own R&D, it would be impossible to drive the kind of scale that makes the foundry economics work. So Ginkgo succeeds in that sense when the synthetic biology ecosystem succeeds. There's no version of foundry biotechnology that can be fully monopolized by any one entity, because we can be good at the DNA programming part, but we can't be good at all of the different products that you can build with biology. We can't be good at cell therapy and gene therapy and making food proteins and agriculture and so on, right? So we rely on partnerships with many, many different developers in order to make the foundry work. And so therefore, it is essential that we make our services accessible and understandable to the ecosystem as widely as possible. So we organize what we do around this idea of services in order to take the very large capabilities, this idea that, well, with DNA you can do anything that biology can do, to organize that into understandable, communicatable services that developers can recognize, can say, okay, I need this particular service in order to make my R&D project work. I'm gonna give some examples of what it looks like when you do R&D at a foundry scale. So what developers can build. And I'm gonna give this example from the biopharma space. I think it's very exciting. I'm very excited about, in particular, all of the emerging modalities in biopharma. They have this common denominator that so many of them are fundamentally programmable. Cell therapy, gene therapy, RNA therapeutics, protein biologics, all of them are written in DNA. And so that means you can systematically engineer them at scale, if you can systematically engineer DNA. And that's true in a way that, for example, small molecule therapeutics are not programmable in that way, right? It's much harder to explore design space for a small molecule therapeutics. How am I doing so far? It's true. It's true. Okay, thank you. Thank you for pushing the buttons. Okay, and I didn't know there was going to be so many awesome talks about gene therapy today. I loved all of those talks. We do work in gene therapy. I think it's a very cool area. I'm taking this example from cell therapy. Cell therapy, I think there's a really stark contrast between the way that existing cell therapies were developed and what the future of cell therapies are going to look like. So if you think about the first generation of CAR T from the 80s and 90s and the second generation, it was done in this very classical genetic engineering way, where you started with a particular biological hypothesis about how to engineer this chimeric receptor. You explored the possibilities, one or a few at a time, right? Just by very painstakingly, manually fusing, okay, I'm gonna take this intracellular domain, I'm gonna fuse it to that receptor, I'm gonna express it in this cell type, and I hypothesize, I hope because my human expertise tells me that the receptor is gonna bind the target, the intracellular domain is gonna control the proliferation and the activation of the CAR T cell in just the right way. And there's just a huge amount of trial and error, right? So this was a decades-long process to systematically find the right intracellular domains. When you do that at foundry scale, you're not constrained by that manual design requirement, right? You don't have to get lucky to choose exactly the right design, because you can explore design spaces combinatorially. Okay, so we took 25 different activation domains, a bunch of different inhibitory domains, co-stimulate, right? Every different modular domain of this receptor, barcoded them, transformed them into 2 different cell types, did a serial challenge assay for 3 different tumor cell lines in triplicate, and we get 720,000 different unique tests in a 3-week period, right? And this is after DNA synthesis, by the way. This doesn't count DNA synthesis, which is still the slowest part. And the upshot of this is that we find domains in this gigantic library that perform better than the classical car domains, right? Because of course they do. Because of course they do, right? Nobody would ever have guessed, nobody would ever have thought that you were going to be able to pick out exactly the right design from the huge combinatorial space. So this is tracking proliferation, persistence of cells in tumor challenge after 20 days for 2 different targeting domains. Because of course you do. So I think in a sense it's sort of obvious that working at this scale is going to lead to better results. But I think what the community is not generally internalized is that this is the way to work, right? Like this has to become the new normal if we are going to be able to generate the most effective, the most consistent therapies. Okay, so big data, automation, lots of DNA synthesis, right? These are the classical components of the bioconvergence. We thought we had it, right? We thought we knew everything. We were the smartest scientists ever, right? Oh, of course, foundries, right? Of course. Then along comes AI, and it changes everything again. AI changes everything again. So I think particularly in this new world of where data drives a lot of biotech R&D projects, we're gonna have to rethink the design again in the context of AI. So where does AI fit into our understanding of how a foundry works? I think the sort of simple version is that AI works together with the foundry and code base in a way that makes data more valuable and more reusable. Right? So AI is a kind of it's an accelerant, right? This idea that accumulating data, accumulating code base is going to create value over time. AI unlocks that, right? It increases the value of data. But I think it goes a little bit deeper than that. I think AI is changing the way that we think about biotech R&D in the sense that it puts the data set that you can generate at the beginning of a biotech R&D project. Often even before the target, even before the goal of the R&D project. You think about the project in terms of the data that you can access to train a model and the data that you can generate. And so Ginkgo is committed to building these data resources, so and data generating capabilities in our foundry and code base, and then making them available to developers everywhere who can use them to train AI models. This is an emerging area. I'm kind of thinking about it out loud here, and I'm happy to talk to the people here about what this looks like. But I think a key unlock in AI is going to be multimodal data. So the ability to accumulate or generate data of many different types in order to train a model for a particular application. So it's not just about RNA-seq anymore. It's not just about metabolomics. It's going to be about all of them together combined for a particular application, right? And so this is my sort of my menu, my data modality menu for a particular app. So I'm going to say, okay, well you're doing a gene therapy. So what do you need, right? You're going to need a bunch of RNA-seq. You're going to want functional screening in different cell lines. You're going to want pooled screening in animal models, cell-based assays. The data set that you need is what is going to enable the application, and in many cases, it is what is going to allow you to discover the right target for your therapeutic modality, for your vector. Okay, so just a couple of examples of this. So this is a data resource that we have. We call this the UMDB, the Unified. Oh no, I forgot the name. We just came up with it. We have a big DNA data set, about 2 billion wild type gene sequences. It's more than, significantly more than what's publicly available. So this is a data resource that we have that we can use to train models on, for example, for protein structure, evolutionary scale models, for example. We have a lot of enzyme data. So we've been working with enzymes almost since the very beginning of the company. We've done more than 400 different enzyme engineering campaigns. Every time we generate an enzyme, we create a library of enzyme variants. We measure the enzyme activity. And this becomes a data resource that we can use to better understand what's possible in enzyme engineering. Boop. Okay. So how do we think about models, AI models, in engineering biology? And as a synthetic biologist, whenever I don't understand a particular new technology, the first thing I do is I make an analogy to tech, to computers. Right? This is our security blanket, right? What does innovation look like? It looks like what is tech doing, right? Let's copy tech. So tech models look like this, right? So you have a large language model, like a chatbot, like a ChatGPT, is trained on a very large library of written text to create a foundation model that captures the broadest patterns that are available in an entire corpus of text. So you have a foundation model, gigantic, often quite expensive to make. On top of that, you have specialized models, application-specific models that take that existing large data set and add to it or do training on top of it in some way in order to optimize it for a particular application. So you have the large language model as a kind of an enabling technology. The applications are much smaller. They're faster, they're cheaper to make, they're project specific, right? So whereas the large language models aren't accessible to every tech company, the application specific models are. I think that we're seeing signs that models for biology are organized in the same way, or that they can be organized in the same way. So the most famous example so far is AlphaFold, of course. There's also a very large data set. There's also a gigantic model that captures everything or almost everything about predicting protein structure. Then on top of that baseline, we have smaller, faster, easier to use, more problem-specific models that are trained on top of AlphaFold as a baseline. And so internally at Ginkgo, we have an enzyme engineering platform, an AI-guided enzyme engineering platform called OWL that works in essentially the same way. So we have a large database of enzyme sequence and assay data. On top of that is the OWL platform. So this is a global platform that helps us design any enzyme. Then on top of that, we have applications that are specific for different properties of an enzyme. So maybe you want to discover an enzyme, you want to optimize the activity, the specificity, the stability, and titer. I think I heard a lot this morning about a particular need for minimizing immunogenicity in engineered proteins. So I don't know if anybody noticed that as like a trend in the talks this morning, right? There's like everyone's, we want to use this engineered protein, right? We need a more active enzyme, but what about immunogenicity? How are you going to evade the immune system? So that's an engineerable property of proteins, probably a good use case for AI. I think it would be a shame if all of the many different therapeutics applications that all need to solve this similar problem all invest the resources independently to solve it. Seems to me a use case where you need a tool that can be accessible for many different applications for solving this common problem of immunogenicity. Okay. The structure of enzyme engineering projects, specifically these AI-guided projects, I think they're, at Ginkgo, I think they're telling us something about what the future of biotech R&D looks like. So this is the history of one particular project, where we took one particular enzyme through 4 different generations of iterative improvement, right? And I won't go over all the details. I'll be happy to talk about it later. But the idea here is that it's a classical design, build, test, learn loop, except now it's powered by AI. And so when I did enzyme engineering as a grad student in the pre-Ginkgo days, these kind of R&D projects had a characteristic pattern when they're led by human insight. And so if you're a grad student and you're engineering an enzyme, your first library is your best candidates. Usually you get the most improvement. Your second library, that's your second best candidates, right? That's why it wasn't in your first library, right? You're moving beyond what is known in the literature, so there's usually diminishing returns. And by the time you get to Gen 3, you're usually not seeing much improvement anymore. AI inverts this. AI changes this pattern, because every time you're generating a new data set in a new generation, you're learning, you're training the model. So you have this characteristic pattern of very large libraries at the beginning of the project to train the model. Then the model gets really good towards the end of the project, and you don't need large libraries anymore. You can work with small libraries. And I think the figure that sort of blew my mind on this, so these are 3 different strategies for enzyme optimization. So the 2 on the right, those are our classical enzyme engineering strategies. And these are violin plots, right, showing the fold improvement that we get for each different variant in the library. It's not just that OWL, the AI-guided design, has the highest performance overall, right? It's not just that. It's that all of the variants are high performance. The whole design space that has been learned by AI is consistently high performing, right? So this is a completely different paradigm for discovery in biotech R&D, right? It's not a haystack. It's not a needle in a haystack problem anymore. This is a haystack of needles, right? There's nothing but needles in there. Okay, so that's, I think, the vision for how we're gonna build these AI models going forward. We've got OWL for enzyme sequences. We wanna do more models. We wanna do an RNA therapeutic model built on an RNA data set. We wanna do cell therapy model based on phenotypic cell data. We are a platform company, so we don't do it by ourselves. We work with partners. And so we are looking for partners in all of these different applications. If you have an idea for a therapeutics application, for example, that would benefit from a gigantic data set, I want to talk to you. I want to build that data set. The bioconvergence changes who can build with biology. Changes who can build with biology. And I think, so this is a big deal. And here I think I'm speaking in particular to the early career researchers in the room, to the young people in the room. The bioconvergence changes who can build with biology. And what I want to offer is this idea that the career that your supervisor might have had, that you might be using as a mental model for how your career will go, how you will create a biotech company, how you will succeed as a researcher, that maybe that model is not actually what the future looks like, right? I think the bioconvergence might change not just what you can make, but how you work, right? Where you live, what your day-to-day life looks like as someone who develops biotechnologies. Talking about the future, so of course it's hard to have complete conviction about anything that involves the future, but one thing that I'm sure of is that a lot of early stage biotech is going to stop looking like this lab, right? So this is the Pasteur lab. It's more than 100 years old, but you all recognized it instantly because this is what labs look like, right? Labs look like big benches, fancy equipment, people, human experts walking around, moving test tubes around, right? That's what a lab is. But a lab doesn't have to look like that, and it doesn't necessarily have to look like that forever. The lab was a response to a particular era in human history. To address the reality of engineering biology, that engineering biology is fucking hard. That it's really, really hard, right? So it wasn't that people loved labs so much. It wasn't that, oh man, I look so good in a white lab coat, right? I really want to go smell formaldehyde every day. The technology of the lab was created because that's what biotech R&D needed, right? It needed a centralized space where human experts could manipulate samples with some consistency, with some reproducibility, with some safety. So that was the technology that the lab solved. The hardness of biology means that we have not traditionally had the luxury in biology of changing how we work, right? You don't get to engineer biology how you want. You have to do it how it wants. The world of tech, I think, again, offers us a sort of an insight to what the future of biology might look like after the bioconvergence, when foundry biotechnology becomes the norm. In the tech space, developers build on top of an existing tech stack. You have simple composable parts at the bottom layers that are integrated, that are designed with in order to build functional systems at the top layers. So biology evolved, it's not engineered, but this similar sort of layers of abstraction model is very useful for thinking about how we build with biology. We have DNA as a kind of a base layer. DNA is organized into genes. Genes produce proteins. Proteins are organized into pathways. Pathways create phenotypes. So there's a similar sort of biotech stack. A foundry can solve the problems, can work at the DNA layer so that developers can build at the phenotype layer. So when you go and work in tech, when you start a tech company today, you don't start by going into a physics lab, going into a chemistry lab, and engineering your own transistors from scratch, right? That would be crazy. You need a specialized space that can handle those, that can solve those low-level DNA programming operations so that you can build more efficiently at the phenotype layer. And so I think the complexity of biology is such that we need to start thinking about this sort of similar division of labor, a similar tech stack around biology, where it isn't the goal of every biotech startup to develop a complete product from scratch. Okay. And so therefore, I think the sort of take-home message, right, for the new developers is that maybe the next generation of synthetic biology startups don't need to focus on physical lab space, right? So in the conventional model today is that, okay, I'm gonna go start a company. What am I gonna do? I'm gonna rent some extremely expensive lab space. I'm gonna buy a bunch of extremely expensive equipment. I'm gonna hire 10 postdocs, and we're gonna work for 10 years doing the same repetitive operations until eventually I can be acquired by a pharmaceutical company, right? So I think in the future, you might not have to do that, and we're already seeing this today. So foundry-powered companies, Ginkgo partners, are able to launch without physical lab space. That decoupling of the physical operations in a foundry and the design and product operations in a developer company is going to change the geography of synthetic biology. It's going to change where innovation happens. And I think I would love for the future of where innovation happens to be Paris, right? Why not? Why not? It makes tons of sense for the future of innovation to be in Paris because talent and product matter more now than infrastructure. What does Paris have, right? Paris has talent, right? I know this. I lived here 11 years, right? I've seen the ecosystem. Paris has talent to rival any city in the world in terms of biological products. Paris is a city that the world comes to, right? If you can work anywhere in the world, if you could start your company anywhere in the world, of course you would choose Paris, right? Yeah, right? I mean, come on, where are you gonna go, right? What Paris doesn't have is abundant, cheap lab space, right? And that's right, and because people want that space for other purposes, right? Maybe it doesn't need it. Maybe it doesn't need it, right? In if the next generation of biotech startups don't need that legacy lab space, now that's a liability, right? That's an expensive investment in an old technology, and we are glad that it was never built in Paris because it would be a waste of space, and it could never be cheap enough to compete with the cheap lab space centers of the world. Right. So this is the future. Oh wait, I had one more. I have a joke to make. Hold on. Wait, let me go back. Okay. See. So look at these. Right. Those are biotech developers. Those are look at them. Look at how happy they are in these photos. That's right. They're happy, having croissants. That was my joke. Arguably, Paris is already the global center of synthetic biology. What? Is that true? Is he crazy? How could you say that? How could you say Paris is already the global center of synthetic biology? Because the iGEM competition is here in Paris. And iGEM is the heart of global synthetic biology. It was in Boston for 18 years. It was in Boston. 2 years ago, it moved to Paris. And now Paris is the city where 5,000 young synthetic biologists come every year. These are the future of the industry. Everywhere you go in synthetic biology, you meet iGEM alumni, right? It's indisputable. There's 20 years of evidence proving that iGEM is the engine, the network of global synthetic biology. And now they come to Paris. All we have to do is give them a reason to stay. Right? Paris is going to be, more than Boston, the city in the world that most synthetic biologists have visited and that they know. So I know about the power of iGEM. This is a plug of iGEM. These are our founders. These are the founders of Ginkgo Bioworks. In 2006, their iGEM project was called Eau de Coli, and they genetically engineered E. coli to make fragrance molecules as a young iGEM team. A couple of years later, they started Ginkgo Bioworks. One of the very first commercial successes at Ginkgo was a project with the fragrance company Robertet. Right, so there's a direct line between the first iGEM project created by the Ginkgo founders to the first product, or I don't know if it was the first, but one of the very early products enabled at Ginkgo, the Eau de Colis project. Since then, iGEM has launched hundreds of startups. There's many, many startups since then that have been associated with iGEM. It makes perfect sense. iGEM teams are given exactly the right kind of training that you need in order to create a synthetic biology company today. So it's a place where I think some of the founder talks earlier today mentioned, right? You have to learn by doing. You have to learn by doing. You have to learn by walking. And that's what iGEM is, right? You throw them out there, that you say, create a product, right? Raise money, talk to customers, understand the human impact on the world. Oh, and also, by the way, you have to do the science. And you have to do it in 4 months so that you can go and show it off in Paris. So it is exactly the right training for entrepreneurship, and that's proven out by the fact that so many synthetic biology entrepreneurs are alumni. And the final example, so this is, I get in trouble sometimes when I say this. I don't know, maybe if French people are, maybe they're too humble. They don't want to hear it sometimes, but I love you. I love you anyway. I think France has a genius for product. France has a genius for product. France is a culture and a history and an industry of creating things that people want, things that the world wants. So things that are beautiful, things that are inspiring, things that are desirable, things that people want to buy. And that genius for product is now going to be even more at the center of the future of synthetic biology, because as the bioconvergence makes biology easier to engineer, the question is going to be less, how do we make it work? And more, are we making something that people want? Are we making something that the world wants? And so I think it would be a shame if France missed out on the bioconvergence, because I think that there's a lot of beautiful things that we can need to make with biology. Boop, boop. And the world needs more biology. And the world needs more biology. And with that, I will say thank you for your attention, and I can't wait to see where you grow. And I'm happy to take any questions. Do we have time for questions?
Are there any questions for Jake? Oh, yes, go, please.
Hi, a beautiful talk, very inspiring. I have one question about Ginkgo business model, because basically you base your whole activity on partnerships, and I see a lot of like different hypotheses. One that are biology-driven hypotheses that you work on, develop different models with maybe data that have been generated first with your partners. And I see also like technology optimization all along for, SynBio, et cetera. And so, could you elaborate more about how do you deal with, like data sharing, what belongs to who, et cetera, and how do you basically use those data that also, like you co-sharing with your partners, and how you move forward with development, because it could be a bit let's say scratchy to have all the freedom to operate in some way.
Yeah, I think that's a good question. So I think that it goes to the importance of being a platform company in this space, and the fact that we don't make our own products, and so therefore we never compete with our customers. The essence of our business model is that we want to succeed when our customer succeeds. And so what that means is that we structure our revenue around success payments. So things like equity that increases in value when the customer succeeds, milestone payments when when they create their product at a particular scale, or other kinds of downstream revenue sharing that kicks in only when the customers succeed, right? So that's important for us because it means we're making a bet on the synthetic biology ecosystem in general. We have a huge motivation to see synthetic biology globally succeed. I think it also it helps with our customers because it removes this kind of adversarial zero-sum negotiation where we're both trying to capture value around a particular technology.
So meaning that you do have all the freedom to operate on the data that you use to build models, is that correct?
Yeah, so that's the short answer. So the short answer is we have to own the IP, we have to own what happens in the foundry so that we can grow our code base, right? So that we can get better at engineering, and then we license the product of a particular program to a customer under the terms that they need to succeed. Yeah.
I think we have time for one last question. No, we don't, but we'll take one. Okay. Oh, there, Barbara.
Oh, now it's red, but it's working. I mean, I guess you must have seen this on Twitter/X already many times, right? This is on the one hand, it's really beautiful. On the other hand, very scary that we're moving on to forgetting about mechanistic understanding and just making something. So you make huge libraries, you have absolutely no idea why that one works or not. It works. And on the one hand, this is good because we want to solve some societal problems, so we might not necessarily care about why this thing is working or not. But biology has always been also about understanding mechanistically. So are we going to be facing a situation where people in the academy, so in university, will work on mechanistic understanding and the companies are going to make products because we can't compete? So if I think about what you're saying, my lab will never be able to produce these huge libraries that you're talking about. So I'm out of the game for making products, right? But I maybe can contribute to the mechanistic understanding. So what is your pick on that?
Yeah, that's a deep question. That's a very challenging question. I don't know if I have a good answer. I don't know if I have a good answer. I mean, I think so. One part of the answer is that we certainly do care about mechanism, right? And almost all of our engineering efforts include some combination of mechanistic modeling and other kinds of modeling. So we don't see, at least in the short term, an idea like this sort of completely black box AI. That's not how we think about engineering biology. I think so. Maybe we're rare in the AI space in that sense because we're not like a pure AI company, right? We weren't created by an AI programmer last year to only do AI. We've done hundreds of enzyme engineering projects, so we do what works, right? And sometimes what works is mechanistic and structural modeling, right? And that's just biology. It's not a silver bullet. The other question I don't know. I think it's fascinating. It's a little bit philosophical. I guess my feeling is that I think this idea that AI offers no insight into mechanism, I think is partially an artifact of it being so new. And I think a lot of new, different kinds. So if you think about, say, mathematical modeling, like a dynamical systems modeling, for example, that's very opaque until you've studied it for 10 years. And then it feels like you have a human understanding of what's going on. And I think AI might play out the same way.
Just one comment. So I have a colleague who said they made a model, so they tried to make a model to predict certain features at the gene level. So some promoters or what, I forgot the details, and he said they had this model that could predict it really beautifully. They spent a year or more trying to understand why, and they could not. So I don't know whether it's true that it's only because we don't know enough about this AI that's why we don't get the mechanism. It depends. So you have to go back and try to do the mechanism understanding. Otherwise, you will not get a mechanism with the AI model. That would be my...
Yeah. Biology does things, and you don't know why. And and so if AI, maybe AI understands it perfectly in that sense.
Thank you so much, Jake. Let's thank him again for this nice talk. So it's time for a round table. So I'm going to invite Anne Jouvenceau and all the other speakers. I'll let her introduce them.
Hi everyone, so we can start? Yeah, it's fine. So I'm really pleased to welcome Christophe Lano, that I know quite well, Stéphanie Alassunière, Jérémy Weber, Kenza Bellage, et Jean-Luc Moulet to discuss on this roundtable about, and I need to read the title that you gave, because it's a long title. It's about how the different institutions of the Île-de-France, so Paris region, scientific ecosystem contribute to the innovation workflow. So we know, and thank you for having said that before, that Paris, and probably France, but Paris as well, is a nice place with a really rich ecosystem, scientific and innovation ecosystem. So we just need to know how we can drive scientific research into innovation workflow, and you will have to explain us how you do. So I think it's really important just to understand that in France we have a lot of different university systems, organization, the SAT, the PUI, and I will not translate that because I'm sure that they will do for you, and the valorization ecosystem as well with Cure Institute and CNRS Innovation. I don't remember the name of your valorization system at the CNRS, but we have a lot of different things, but you will see that everything is quite well working all together. So if I may, I would like to start with Jean-Luc. So Jean-Luc Moulet is, and I have my anti-session, is the Deputy Director of Innovation at CNRS, is that right? And when we prepared the roundtable, you just say that innovation arises from scientific projects and collaboration. So can you tell us a little bit more about that and say how CNRS deal with that?
Okay, thank you. Thank you very much for having me today. Very pleased to be here. So we do represent today 5 different research organizations, and despite all the differences we may have, we have something in common, which is basically almost the same processes. And more importantly, and thanks to France 2030, we have money to help jumpstart startups and help company to be built. The primary issue I think we face is that we are lacking something in a significant manner, which are projects. If we compare the potential of what we have in France in terms of academic strength and the number of startup projects we build on a yearly basis, there is a gap. And I guess our main challenge would be to fill that gap and to be able to identify more projects and build up more startups. We can work with that using the usual tools of communication, symposiums, talks, shows, etc., etc. But at the end of the day, I think we tend to convince people who are already convinced, and we